So furthering the promise of personalized medicine, here at City of Hope, Joseph Gold directs manufacturing at the Center of Biomedicine and Genetics. Now, you know about those pluripotent stem cells? He's been working on them for 30 years. He's been bringing in disease modeling, cell-based therapeutics, and oncology applications to that work. And among the diseases the center is tackling cancer, heart disease, macular degeneration, ALS, you know, the easy stuff. He's here with it.
Okay, I have to admit I was really nervous about giving this talk because I've never done one in this particular format before. And then this morning, it suddenly occurred to me that this is actually the perfect format for me because A, I talk too fast, and B, I pace too much. So I think it's going to work to my advantage here, but we'll see. So far, you've heard from some really innovative scientists who have developed novel therapies to treat a variety of diseases, and you've heard from clinicians who are applying those therapies in clinical trials. But I have a different job. I make weapons. I'm the Director of Manufacturing at the Center for Biomedicine and Genetics at City of Hope, and CBG is what I define as a biological forge. And by this I mean that we take those prototypes and figure out how to mass produce them at a scale and at a level of purity which is appropriate for use in human beings. Now, we're dedicating our efforts to building up an arsenal against a variety of diseases, which you heard Pat mention in passing. I wanted to just touch on a few of the weapons we're building here because you've heard about some of these already. We're making lentiviral vectors for CAR T cells, and you heard some very eloquent presentations about how important that is. We make these vectors. Some of them have been designed by City of Hope PIs, like Dr. Brown and her colleagues. Some have come from external investigators or other companies, and CBG's role is to make very large quantities of these vectors to supply for the clinical trial work, and some of these trials take place at City of Hope, some take place externally. But that's not the only kind of oncology project we're working on, and I wanted to actually highlight what I think is a really interesting approach which is being taken by Dr. Karen Aboudi here at City of Hope, because she's come up with another way to treat glioblastoma, which, as we heard, is a very, very difficult disease to treat. And her approach is a combined cell and gene therapy approach, and she starts by working with neural stem cells derived from fetal brain. And these are cells which normally would play a role in the development of the brain, but they also have an intrinsic capacity to migrate to sites of brain tumors. So Karen is using these cells as a delivery mechanism, and she's taken them and modifying them with a viral vector that makes them express an enzyme which converts an inert chemotherapy drug into its active form. So what she can do then is inject this cell therapy into the patient, and it migrates to the sites of the tumors, and then she can give the inert chemotherapy drug, which has no toxic effects on the rest of the body, but it's converted to the active form at the site of the tumor. So it's a very neat way of making this delivery. And Karen's approach differs from the CAR T methodology also because while CAR T is an autologous product, meaning you have to produce an individualized batch for every single patient, Karen's using a cell line which can be used in multiple different patients. So we can make up large banks of these cells and freeze them and then thaw them when they're needed. And CBG's role has been to mass produce the adenoviral vector that she's using to modify the cells, and also to help her group create large cell banks of these therapeutic cells. And what I'm pointing out here reflects the fact that we do a lot of work at CBG with human embryonic stem cells and their closely related human-induced pluripotent stem cells. And these cells, of course, have the really remarkable capacity to be converted into any type of cell under the appropriate conditions. And here what I'm showing you is that CBG has come up with a method of producing very, very large quantities of cardiomyocytes, the heart muscle cells from these kind of cells. And these are being tested in animal models of heart failure, and we hope they're going to prove effective, and they may also be useful in attempts to repair neonatal heart defects. And this is important because this is an example of how the team at CBG not only figured out how to do it efficiently, so we can make batches of cardiomyocytes that are 95% pure, but we can also do this at a pretty large scale. So we're generating billions of cells at a time, which is important because this particular application will probably require hundreds of millions to billions of cells per patient. So we're tackling a lot of different things. We're using a lot of different approaches to tackle a lot of different diseases, as I mentioned, and these things include, in addition to these, we're about to start a clinical trial using neural stem cells modified in a different way to treat ALS. And this is a phase 1 trial that's starting at Cedars-Sinai under the direction of Dr. Clive Spencer. We are also in the middle of a clinical trial where we've produced retinal pigmented epithelial cells from embryonic stem cells, and these are being used in a trial to treat macular degeneration. We're about to wrap up to do some work to treat some devastating childhood diseases like Canavan's disease and Hirschsprung's disease using stem cell-derived therapies. And we're also beginning to work on diabetes work and some Alzheimer's work, et cetera. It goes on and on. So we are really trying to build up a large arsenal of weapons. I can't make this go forward. Can somebody do this for me? There we go. So there's really something important to keep in mind here, which is that any weapon or any therapy can be dangerous if it's made or used badly. And we've had a lot of examples of this recently. Some of you may know these stories. In 2016, there was a report of a 66-year-old man who started suddenly having back pain and trouble walking. And he was examined at Brigham and Women's Hospital in Boston, and they did an MRI of his spine and found the lower spine was filled with some kind of mass. And they opened him up, and it was a very odd-looking, primitive-looking tumor. And when they took some of that tumor out to analyze it, they were really shocked to find that it wasn't even derived from his own body. So how could this have happened? Well, it turns out that back in 2009, this man had had a stroke, which left him with some motor deficits. And he saw an ad for a clinic, which was not FDA regulated, that advertised a magical stem cell therapy that could repair stroke damage and treat aging and heart disease and on, you name it. So he went to this clinic and was referred to other clinics in Mexico and Argentina and China, where he was charged tens of thousands of dollars for multiple injections of this unknown stem cell product. And the end result is what I described to you. He's got a tumor growing in his spinal cord they don't know how to stop. And this is not the only example of something like this gone wrong. Just last week, there were stories about 3 patients who were suffering from macular degeneration, and they went to a clinic, not in the third world, in South Florida. And they went to a clinic which, again, did a process which was not FDA regulated, and this clinic extracted stem cells from their own fat tissue and then injected it into both eyes in each patient with this experimental therapy. And the end result is that these 3 patients are now blind. So certain things are simple, right? I mean, I think we would all agree that what happened to these desperate people is a tragedy. And I think it's not a stretch to say we're all in agreement that it is morally reprehensible that anyone would take money from desperate people and inject them with a dangerous therapy like that. But what may not be so simple or obvious is how do you make sure that the therapy that you're making is safe and efficacious? Well, there's really not a lot of shortcuts there. If you're going to do it right, you do it according to current good manufacturing practices, and you'll see that abbreviated as CGMP or sometimes just GMP. And what this really means is that, first of all, you exhaustively vet the raw materials you're going to use. Secondly, you are incredibly compulsive about following very, very detailed protocols to make the product. And then finally, you extensively test the final product to make sure it's exactly what you think it is and not contaminated with anything that shouldn't be there. So how does this really work in the real world? Well, any successful manufacturing facility like CBG works because of a dynamic tension between the manufacturing and the quality groups. And it's very important to note these are independent groups, and that's important because manufacturing's job is to figure out how to make the therapy and to produce it, whereas quality's job is to make sure that all the appropriate standards are adhered to. And there can be differences of opinion between these groups, but ultimately, we come to an agreement, there's an alignment, and we work together such that what we make is as safe and efficacious as we know how to make it. Now, take, for example, how this would work. If we talk about the raw materials, there are hundreds of companies that sell you thousands of different reagents which are perfectly appropriate for use in basic research. But it doesn't mean that any of those reagents are safe to use in human beings. And I'm going to give you actually a really complex example. I mentioned the work we do with human embryonic stem cells. Well, the original stem cell lines were isolated back in the 90s by James Thompson, and he did this by really complex manipulations on microscopic early human embryos, and they were treated with things that included factors for pig serum, and those cells were then plated down on a layer of irradiated mouse cells to keep them attached and alive, and these cultures were then fed with media that contained 20% fetal calf serum. And that's what you had to do back then. We have better methods now, but it worked. And those cells have been used worldwide and have taught us an incredible amount about human development and diseases. But there's a huge leap to take those cells from that research application and put it into patients. Think of those materials I talked about. What if the pig factors came from animals that were infected with porcine viruses, or the mouse cells had murine viruses? Are your human cells now infected with these unusual viruses? And the fetal calf serum, what if it came from animals that had mad cow disease, which may sound like a kind of esoteric or weird thing to fixate on, but if you recall back in the 1990s in the UK, there was an outbreak of the human form of this disease from people who had eaten contaminated products that were contaminated with prions, which are not even viruses, they're an infectious molecule, which are very, very hard to detect. So what do you do about this? The answer is, if you're going to be using cells in GP manufacturing, you have to screen them incredibly intensely for basically every pathogen you can think of. And if you're going to work with these cells and using various reagents, you try to avoid certain types of reagents. You'd always prefer to work with synthetic or recombinant molecules if you can. But if you have to use something like fetal calf serum, and sometimes you just do, you make sure that it comes from animals which have been maintained in specially monitored herds in places like New Zealand, where there's no endemic mad cow disease. And just in general, any reagent you use in manufacturing, when it comes into our facility, is quarantined until we know it's exactly what it is, and it's ready to be used, and it's safe. And then we track very carefully exactly when and where it's been used, so everything's traceable. And with regard to the actual productions, it's not just that, of course, we make sure all the equipment is carefully maintained and calibrated so it's working exactly the way it's supposed to. And it's not even just that we follow these extraordinarily detailed SOPs and don't deviate from them at all. It also includes things like, it's not just the operator doing the operations, there's literally someone called the verifier whose job it is to stand behind that person with a checklist and make sure every step is followed, every bit of the way. And even when the product is finished, it's not released for use in humans until it's gone through a whole lot of extensive testing, again, to make sure it's sterile, to make sure it's pure, to make sure it has the biological activity that it's supposed to have. In the case of the CAR T-cell vectors that I mentioned earlier, those vectors undergo about 17 different tests to make sure that they're sterile and that they're exactly the right virus and not something else. In the case of the cell therapies that I've talked about, those undergo more than 24 different tests to make sure that they're sterile and they're not contaminated with any cells you don't want to be there, and to make sure that they have the right kind of activity. And, to give an example, these standards are really, really rigid and inflexible for a good reason. We have produced a particular batch of a cell therapy that had 1% lower biological activity than our standard called for, and as a result, that was rejected for use in human beings. That's how strict we have to be on this. So any good manufacturing facility succeeds, because of its fixation on the materials and the process and the product and the testing. And, right now, I realize that there's a lot of talk about how onerous regulations are and how things are moving too slowly and that we should be able to speed things up. And it's absolutely true that every system can be improved. That's just human nature. But until somebody can convince me that all raw materials are perfect, that human beings are infallible, and that every process works exactly the way it's supposed to every single time, then facilities like CBG will continue to do our job. Okay?